Thanks so much for joining me for another edition of Parker Biology. Today we're going to be looking at cell communication. We'll be thinking of examples of how cells communicate, why they would want to communicate, and exploring the mechanisms that allow cells to talk to one another. Take a minute and pause this video and talk to your neighbor about how cells illustrate the unity and diversity of life. How do they show that life is united, yet there are unique features as well? Take a couple minutes and talk that over. Your answers will vary, but if we look at things like similarities in structure and function, that can really help illustrate that the basic building blocks are the same across domains of life. We're looking at phospholipid bilayers forming membranes, which help facilitate control of passage into and out of cells. We're even looking at the idea that all eukaryotes have nuclei and other membrane-bound organelles. All of this put together does point to the idea of common ancestry or divergent evolution, that everyone branched off of a similar ancestor. What we'll see throughout this unit is that communication pathways are also very similar. Cells talk to each other in a very similar manner, even though they might be talking about different things. This again is going to help point out this commonality, this unity of life. Next, we have two examples of communication between cells. The first is communication between mating yeast cells. There are two different types of yeast cells. One is called the type A, and it releases what's called an A factor. The other type is called an alpha yeast cell, and it releases the alpha factor. These factors don't enter the other cells, but they do bring about changes. When the alpha factor binds to an A cell, and when the A factor binds to a neighboring alpha cell, they bring about changes that allow these cells to grow closer together and to fuse. And upon fusion, the new cell possesses all of the genes of both of the original cells. This new cell will possess both the A and alpha genomes. Take a minute with your neighbor and talk about why this would be an advantage to the yeast cell. How would this help out to have two sets of instructions? Go ahead and pause the video and discuss. Why would it be so helpful for a cell to have multiple sets of genetic instructions? Well, more genetic material means this organism is going to be better equipped to deal with any potential environmental changes. If the environment shifts in one direction, it may have the genetic cards to play to help it win. This also means more possible combinations for the descendants. The more genes you can pass on to your offspring, the more the offspring benefit, and the better overall that is for the species as a whole. Based on this example, we can see that mating is a great reason for cells to talk to each other. But even asexual prokaryotes need to talk sometimes. Bacteria can communicate with a method known as quorum sensing. And it's through quorum sensing that they can monitor how densely they're populated. So a single bacterium by itself is probably not going to launch a huge pathogenic attack against you. It's going to take millions of cells to be able to do that. So the bacterial cells can talk to each other so they can sense how many bacteria there are, and they don't launch the attack until there are plentiful numbers. In addition to launching a full-fledged attack, sometimes bacteria talk about forming biofilms. And this is a collection of cells that stick not only to each other, but also to a surface. And they couldn't do this unless there were many cells working together. These biofilms are evident when you wake up in the morning and you have that film across your teeth. This is a result of bacteria dividing throughout the night while you sleep. Whether cells are talking about mating or communicating their numbers so they can launch attacks or form biofilms, it's clear that cells do need to discuss things with each other. And the next big question is how? How do cells communicate? The ability of a cell to respond to a message is determined by whether or not it has a specific receptor molecule. It needs to be able to bind to a signaling molecule. So when a cell talks to another cell, it sends out a signaling molecule, and if the receiving cell has the right receptor molecule, it can hear the message. When the signals are received, they're changed, they're transduced, and ultimately, we should see cellular response. The cell hears a message and then it does something about it. And the series of steps that this goes through is known as a signal transduction pathway. We'll be looking at several examples of receptors, we'll look at how exactly transduction works, and we'll see options for cellular responses. This next series of diagrams will allow us to look at different types of signaling. What we want to do is actually match up these different pictures that we see in the model with these 4 key terms off to the side. Go ahead and analyze pictures A through D and match up the different symbols that you see. Pause the video now and do this, please. Hopefully you noticed in pictures A through D some commonalities. We can see this small, almost soccer ball shaped structure that's being passed from one cell to another. So that's the signaling molecule, or the ligand. Here's the cell. Here we see a membrane channel. And here we've got the receptor. And the next question wants us to describe how the shape of the ligand and the shape of the receptor are related. What do you notice about the two? Good. The shapes are complementary so that the ligand should fit into the receptor. And that specificity and that complementarity is really important in ensuring that only certain signaling molecules get to bind to certain receptors. Take the next few minutes and on your own, using pictures A through D, answer numbers 3 through 7. Pause the video now, please, and answer 3 through 7. Number 3 asks, which type of signaling is for cells that are in close proximity to one another? And the best choices here are B and C. Choice A is actually signaling to itself, but B and C are both relatively close to one another. For question 4, it wants to know about cells that are in different parts of an organism, or long-distance communication. And our best choice here is D. Notice on choice D, it looks like that ligand is having to travel through some sort of vessel, perhaps the blood. That brings us to number 5. In which of those would the ligand, or the signaling molecule, need to have the longest life? Good. D would need to be the most stable, or have the longest life, because it's going to need to travel longer distances. If it's degraded too quickly, it won't make it to its destination. Next, we're being asked to give pictures A through D names based on these word part definitions. So first we're being told that autocrine, auto means self. So which of these pictures best describes a cell talking to itself? Good, your best choice here is picture A. Next, you're being told that juxta means beside, next to, or most specifically, touching. So where do we see two cells talking that are touching? Good, that's picture B. So here we've got juxtacrine. Next, paracrine. Para tells us nearby. And here we already said that cells in picture C are fairly close to each other. So we'll make this one paracrine. And that leaves picture D to be endocrine. And when we match it up with that word part within, we're looking at two cells within the same organism. And this is usually characterized by that signaling molecule moving through a blood vessel. As we can see, there's several different types of signaling, whether the cell's talking to itself, a cell that it's touching, a cell that it's close to, or just another cell within the same organism. We can also see that communication can take place for a number of reasons. Mating, like in the yeast, defense mechanisms, homeostasis, growth, like in forming the biofilms that we talked about with bacterial quorum sensing. So do all the ligands for communication have the same shape? Clearly not. If they did all have the same shape, all of the signaling molecules would bind to all receptors, and all the messages would just get misunderstood. Now, to see if we truly understand these different types of signaling, I want you to read through the following examples of cell communication and determine which type of signaling, autocrine, juxtacrine, paracrine, or endocrine, each example or scenario is trying to describe. Please pause and continue on each of these examples. You'll want to continue up until the Roman numeral 4. Let's double check ourselves on these scenarios. So first, we've got plasmodesmata. We've seen these before. Remember that these are channels that connect plant cells so that materials can flow between them, including signaling molecules. Since plasmodesmata are characterized by cells touching one another, what's our best type of signaling if they're touching? Good. This is a great example of juxtacrine signaling. Next, we've got the morphogens. And morphogens should look a little bit familiar. These are examples of cytoplasmic determinants, helping developing cells determine what they're going to be. And here we can see these cells are close but not touching. So which type of signaling is best for close but not touching? Good. We should call these a great example of paracrine signaling. In our next example, we see cells in the pancreas talking to cells in the liver. And these cells are not close by. They're going to rely on hormones like insulin to move through the blood between them. So this is going to be a great example of endocrine signaling. Our next scenario looks at cancer cells, and it says they release their own growth hormone. So they're talking to themselves. What is this an example of? Good. Talking to themselves is going to be autocrine signaling. Next up, we see neurotransmitters released from the axon of one neuron, traveling across a very short synapse and binding to a dendrite of another neuron. These are extremely close together, but not quite touching. So we'll call this paracrine. And finally, cells in the pituitary gland produce thyroid-stimulating hormone. This is received by cells in the thyroid. So notice this is movement between the pituitary gland of the brain and the thyroid found down in our neck. So since this is not close and this does rely on hormones traveling through blood vessels, we're going to again call this one endocrine. Thanks so much for joining me for this edition of Parker Biology, where we explored types of signaling. In our next video, we'll be looking at the discovery of signal transduction pathways along with their mechanisms.